Right. Hi, guys. Hope you've been keeping okay, and enjoyed your well-being day last week, and are managing everything. All right. So today, what I'd like you to do is have a look through this video, and I've got the PowerPoint as well for you. I put myself up here, and I'm going to talk through Hardy Weinberg. Okay, and we're going to see whether or not we can get you to really understand this. You've also got a document with questions. Type in the boxes. Okay, that will give me a bit of understanding as to your grasp of this. All right. Now I was going to do. I'm not too sure what's going on there. I was going to just set you. I searched online for a video explaining this, and frankly, I didn't think they were very good. So I've decided, nope, I'm going to do my own for you guys. So I'm sorry. I know it's me again, but that's life. Right. Okay. Gene pool, allele frequency. So you must go back and remind yourself what an allele is. Yeah. So different form of a gene. Hopefully, we spent a long time going through and doing the inheritance in school last term. Hopefully you're happy with alleles, dominant, recessive. We're not getting into anything complicated here. There's no dihybrid, and in fact, really, you can only use this principle on a single, gene with can only have two alleles. Okay, and it must be a simple dominant recessive. No codominance, nothing like that. It doesn't work otherwise. So. Let's run through this. We've got populations, ecosystems. Gene pool is the whole population with all of the alleles for a particular gene. Okay, and here we've got this diagrammatical representation of our mice. Okay, the dots inside the mice are representing the alleles. Okay, and so we can see there, I'm covering one up, that we've got what must be 10 mice, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 mice, 20 alleles. Okay, you can count the black alleles, and you end up with 12 out of the 20 possible alleles. 10 mice, 20 alleles. They each inherit one allele from mum, one allele from dad, so each mouse has to have two. 12 of the 20 possible alleles are for the black dominant allele, and that leaves us with 8 for the white recessive. The problem is, when we look at organisms, you cannot tell. Okay, here, if this black coated mouse is heterozygous or homozygous dominant. Okay, they both look the same, so there's no way of knowing. Yeah, you cannot physically, test the entire population, and we're talking populations of hundreds of thousands. Yeah, so it wouldn't be efficient anyway. However, you can identify. Look at this: the recessive, the homozygous recessive. That will always be the same, and that's why you can only really use this in a limited capacity for single gene disorders. So you can always count in a population the frequency of this genotype, okay? And then we can use that to work out the others using our equations, okay? Again, this sort of statement is this idea that an allele frequency will remain the same as long as certain conditions exist. Okay, we listed those. These are slightly different to how I would have told you them in class. Might be slightly different to how they're written in your textbook. But ultimately, you need to remember a couple of these. I always say that the one I remember is random mating. Okay, the mating must be random. The population must be large, in the thousands, not in the hundreds. Okay, then we've got this idea of you don't want any migration or bringing in of new alleles, in and out of this population. So you're sort of saying a stable population, and it says no mutations must occur. So random mutations to these alleles that would affect them, or bring in a third allele, that sort of thing. And lastly, all the genotypes must be equal. What that means is that one genotype, well, actually, more technically speaking, one phenotype, that's the result of the genotype, is not selected for, okay, positively, or selected against negatively by natural selection. Okay, so basically saying that both genotypes that are possible have an equal chance of survival, and as long as all of these are met, then it's saying that these population frequencies ought to remain the same. Right. So this PowerPoint does it as a Punnett square, which I don't normally do, but again, it's good to look at things in a slightly different way. Yeah. So here, if we get this is deriving our two equations. That's all right. I can stay up there. How do we derive our two equations? Well, like we said, you're given the equations, so you don't need to learn them, but you do need to know what they mean. Okay, so we've got this here, the P and the Q, and you've got to remember that P is dominant, Q is recessive. You do have to remember that. Remember, I said dominant D is before recessive R in the alphabet. And P is before Q, so you might be able to remember it that way. And so we know that in a population, all of the alleles that are present, whether you have the dominant or the recessive, all of them together will equal 100% of your population. Yeah. So every single person that you are counting, including in your population, their alleles all of them will add up to an equal 1 or 100%. Then if we took here two heterozygotes, okay, a PQ, a PQ, and we look at the potential offspring that we can get, we can get P multiplied by P, so P squared for this genotype. We get PQ and QP, B, big B, little b, big B, depending on, you do have a little b allele there and a different little b allele there. So you do have two of those. So you end up with two lots of PQ, okay? And then your little b, recessive, homozygous recessive, they've done that wrong, haven't they? That should be little b, is Q squared, okay? And that's how we end up with this equation here, yeah? And again, we know that for all of these four possible, or really it's three, isn't it? Genotypes, that those also must add up to 1, okay? Because again, every single person in the population you're counting has to be either that or that or that, yeah. And so with our mouse population, you could use the numbers that we had that we'd calculated as 0.6 for our value of P, 0.4 for our value of Q, and remember we just have to square them, 0.6 squared, 0.4 squared to get our two alleles, and then our two PQ in the middle. And what we would find is that they always add up to 1, or they should, if you've done it correctly. So let's run through a couple of examples. First of all, we've got our pigs. Okay, we've got our pink coat being dominant, a black coat being recessive. Okay, it's impossible to count the allele frequencies from the phenotypes. Like we said, you can't see. So some of those pink pigs will be heterozygous, okay, dominant and recessive, and others will be homozygous dominant. Yeah. So how do we find those out? Well, we always start with the homozygous recessive, in this case, the black coats. Okay, so we count those and we end up with 4 out of our 20, I think it is, 2, 4, 6, 8, 10, 12, 13, 14, 15, 16, 16. So we've got 4 out of 16, okay, equaling our proportion, our frequency of homozygous recessive. 4 divided by 16 gives us a value of 0.25. Now, do remember, when you're using these populations, you are calculating, you're identifying the genotype. Okay, so it's 2 alleles. Yeah, so it must always be, almost always start by identifying one of these values down here in this equation. Okay, yeah, very rarely do you start a question and you can figure out P or Q on its own straight away. Normally, you identify like we said, the homozygous recessive, that frequency, and then we work backwards. Okay, so here we've identified that our homozygous recessive, proportion is 0.25, 4 out of 16. So then we can square root that, okay, to get Q, a value for Q of 0.5. Since P plus Q always equals 1, we know that 0.5 plus 0.5 will equal 1, so we know that P must be 0.5 as well. 0.5 plus 0.5 equals 1. Then we can use that to calculate our heterozygotes. So we can do 2 times P times Q. So 2 times 0.5 times 0.5, okay? Which means that we end up with a value of 50% or 0.5, okay, of our pink population will be heterozygous. So if we've got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 pink pigs, then 6 of them ought to be heterozygous. Okay. If I went through that too quickly, go back, rewind, watch it again, work it through, pause it, listen to what I'm saying. When I'm saying homozygous recessive is this, or square rooting that, or whatever, and really take the time to try and understand this. Okay, here we've got just a slightly different way that they can present the information to you. Here we've got our fruit flies, okay? Got a population of 1,000 fruit flies. Okay, 640 have red eyes, while the remainder have sepia. The sepia eye trait is recessive to the red. So how many individuals do you expect to be homozygous for red, the red eye colour? Well, first of all, we need to identify our homozygous recessive population. So here, they have not counted it for us. They haven't counted the recessive. The sepia eye trait is recessive, but they've only told us the number with red eyes. But what we can do is we can figure out, if 640 have red out of 1,000, then 360, the remainder, must have sepia eyes. Yeah? Do you understand that, hopefully? So we can then take that, 360 divided by 1,000 to turn it into a proportion. So 360 divided by 1,000, 0.36. That's our value for Q squared, our homozygous recessive. We then square root to get Q. We do, what 0.6 plus what will equal 1. What must P be? So P must be 0.4. Then we can do P squared to find, because we've been asked to find the homozygous red, homozygous dominant. P squared, 0.4 times 0.4, 0.16. Okay, so you get that. And then if they're saying how many individuals, then remember you've got to then convert that back into, that's 0.16 out of what are 1,000 individuals. So again, just multiply by 1,000. You divide by 1,000 to turn it into the proportion. Multiply by 1,000 to turn it back into the population. But it could be any population size. It could be 10,000. It could be 100,000, 150,000. Okay, it doesn't matter. Yeah. And then you would get the number of individuals. Right. Uses of this, okay, here. So this is talking about a bit more of a real life situation. We've got PKU, which you probably don't remember. We do discuss it very briefly in a lesson in year 12. It's an enzyme, single gene enzyme disorder, whereby you're unable to make phenylalanine, one of the amino acids. Okay, and if it's not picked up quickly, then in children who have this disorder, then they develop brain damage very, very quickly. Okay, and it's very simple to treat. You just have to give them supplements, give them enough phenylalanine in their diet, and then they're absolutely fine. They develop normally, but you need to identify them. So here we've got a patient's child. He's a carrier of the recessive mutation for PKU. Okay, the parent of the child wants to know the probability of her grandchildren inheriting the disease. Okay, so you've got a grandmother who's got a child. Yeah, well, not a grandmother yet. Mum who's got a child. That child is heterozygous, so they know that they're a carrier. What's the proportion? What's the chance of them meeting someone and having a kid who will then have PKU? Okay, we don't know, like it says here, we don't know the mutate the allele frequency. Okay, yeah, we don't know how many other people have that, but we do know the disease frequency. Okay, so people who are born with it, they're recorded, so we have that. So we can work backwards from that. The homozygous recessive, the people born with PKU, test positive. We know their allele combination, their genotype. And so what we can do is work back to then figure out how many people in the population will be a heterozygote. Okay, like it says there, heterozygote who might, if they then meet together and they reproduce, produce a child with the homozygous recessive. So 1 out of 10,000 babies is born approximately. So that's our data. That's our value. So we go, okay, 1 out of 10,000. 1 divided by 10,000 will give us our proportion, our number for Q squared. Okay, our recessive, genotype. Okay. Using the equation, predict the percentage of human population will be heterozygous. So once we identify here 1 out of 10,000, 0.0001, we've just divided 1 by 10, 1, 2, 3, and 1,000. Yeah, we get our Q squared value. We then square root that to get our value for Q. We then take that away from 1 to get our value for P. Okay, then we can just use our second equation, 2pq, 2 times 0.99 times 0.01 to give us a value of 0.0198. Okay, that's our proportion, for 2pq, or our heterozygous, in this case a carrier. Okay, and then if we want to turn that into a percentage, multiply it by 100, 1, 2, move it to 2 spaces, or if you wanted to get that. So here it says predict the percentage. So you've got to read the question. 1.98% of the population, or you could do it out of your population, if they gave you a population. 1 in 10,000 babies, but you've got a population of 200 million. You could then, do 1.98% of 250 million, whatever I said, to get a number of people. So I really hope that that's gone through it. It's a bit quick. Watch it, take your time, have a go at the questions, type in your answers into the boxes and hand them in. We'll then be meeting at the second half of the double, okay, where we can talk through this to double check and answer any questions, check any misunderstandings, anything that you guys are still working on. All right, well done. Right, how do we stop?